Chapter 10 focuses on DNA as the chemical nature of the genes. So we will first discuss very briefly the history of study of DNA, how DNA has been identified as the source of genetic information. Afterwards, we will talk about DNA structure and discuss the relationship between the structure and the function of DNA. The first question I would like to ask is, what are the key characteristics that genetic material has to possess in order to serve as a blueprint for constructing or for coding for all form of lives? Look around you. You see, compare human being, trees, bugs, animals. You see tremendous diversity around you. However, the coding instruction in all of these individuals are the same, are written in the same genetic language, in the nucleic acid by encoded in genetic code. So, therefore, since this language has been preserved throughout the evolution, it must possess important characteristics. So, what are these characteristics? First of all, genetic material must contain complex information. It has to be capable to store a large amount of instruction. Remember that our DNA, our genetic material, is our blueprints, which contains code of life. It defines who we are. So it really has to have this capacity to store huge amount of information. Secondly, genetic material must replicate faithfully. Imagine we all start as one cell, fertilized egg, zygote, and the zygote, one cell, replicates billions times. There are billions of cellular division. Imagine if the replication is not faithful. It would end up in many deformation, malformation, diseases, and so on. So genetic material must evolved mechanism which assure that replication is faithful. Third thing, genetic material must encode a phenotype. So the code, chemical code, has to be translated in something which encode phenotypes, and we know that DNA encodes mRNAs, RNAs, and RNAs, and afterwards translates the code from DNA into proteins. And the last one, the fourth one, which is not outlined in this slide, is genetic material must have the capacity to vary. You recall, variability is very important for evolution. If there was not genetic variability, there will not be evolution. So let's now discuss very briefly the basic discoveries which contributed to nowadays understanding and knowledge of how DNA encodes genetic information. And we have to go more than 100 years back to understand the road which was taken up to the time when we could actually define the structure and function of DNA. First, I would like to point at Miescher's discovery of nuclein. Miescher in 1869 published a result of his work where he studied a pus, pus, that ugly white thing which appears when we cut our cell and it heals sometimes. And basically the pus is composed of white blood cells, which are good bodies because they are called to the site where pathogen penetrates and defend us against the infection. And once they finish their work, they die. So what is so interesting about the pus? Those white blood cells have large nuclei, and Miescher developed a method how to isolate this nuclei. Once he did so, he identified a material within which we call nuclein, and we know that it is nucleic acid. So he did discover nucleic acid. Now, at that time, around and towards the end of 19th century, many different laboratories identify nucleus as a physical basis of heredity. However, the question which becomes the controversy among them was, is it nuclein or is it protein which encodes genetic information? You know that nucleus contain both nucleic acid and protein. And basically there was question and dispute which one encodes genetic informations. At that time, Mendel's work was published. However, it did not trigger too much of interest, and also he contributed enormously to our understanding how the traits are inherited, and his law and principle are valid up to today. His work didn't put any light onto what those factors and phenomena which he describes are. So his work didn't brought any understanding towards the molecular mechanism by which genetic information is inherited. Now, at that time, at the end of 19th century, I would like to point at one discovery which contributed enormously to our understanding of nucleic acid, and it was Kossel who determined that DNA contains four nitrogenous bases: adenine, cytosine, thymine, and guanine. So basically, he was first one. So nitrogenous bases were discovered already in 19th century, and this was huge progress. However, based on his discovery, Levin at the beginning of 20th century postulated or proposed tetranucleotide theory, which actually pushed back understanding of DNA as a material which encodes genetic information backwards. What does it mean? In the tetranucleotide hypothesis, states that DNA consists of series of 4 nucleotide units, and each unit contains all 4 bases in fixed sequence. So basically, if you imagine that, what would it mean that every organism has the same genetic information? It could not explain variability within genetic information. And actually, this hypothesis pushed forward the idea that protein, where you have 20 different amino acids which build them, might be a structure which are variable enough to encode genetic information. This tetranucleotide theory has been disproved by Chergaff only in 1948. You see, almost middle of 20th century. And what he found was that he isolated and analyzed nucleic acid from different organisms, and he observed great variety in base composition. So if you apply tetranucleotide theory, this could not happen. So basically he disproves this hypothesis, tetranucleotide hypothesis. In addition, he actually formulated what we call Chergaff rules based on some observation he made. And what these rules and his observation actually states are that the amount of thymine always correspond to the amount of adenine, and the amount of guanine always correspond to the amount of cytosine. So purine, pyrimidines, Chergaff rule. Now, at the beginning of 20th century up to the mid-century, there were several different experimentation and results published which actually proved that DNA is a chemical nature of the genes. And we will discuss a little bit in more detail Griffith's experiment, transforming principle, and Avery McLeod experiment, which actually identified what the transforming principle is, that it was DNA, and final proof, which was given by Hershey and Chase, that DNA is genetic material. And of course, we will talk about Watson-Crick's model of DNA. We will now discuss sets of experiments which provided evidence that DNA rather than protein was a genetic material. Before we go there, I want to point out the fact that at that time, at the beginning of 20th century, it has been already known that chromosomes are carrier of genetic information. However, as you know, chromosomes compose of DNA and proteins, so the question which one is carrier of genetic information was not solved. The first clue that DNA was a carrier of genetic information came from Griffith's experiments. He first defined transformation in bacteria and he described so-called transforming principle. Griffith was interested in Streptococcus pneumoniae and infectious bacteria which causes infectious pneumonia. He worked with a strain which he called 3S, which was virulent pathogenic strain. This strain contained a polysaccharide coat and produced very smooth colonies while grown on agar. Occasionally, however, he observed and he isolated also non-virulent strain type 2R, which lacked polysaccharide coat and produced rough appearing colonies. So he could very easily identify virulent and non-virulent bacterial strains while growing them on agar. Griffiths was very interesting in the nature of the principle or nature of the substance which is responsible for the pathogenesis. So he picked up his studies is two virulent and non-virulent strain in mice, and he asked the question, what is responsible for the pathogenesis? So, of course, when he performed his experiment, he grows the bacteria 3S and 2R, virulent and non-virulent, and he injected the bacterial preps into mice. These were his control. So of course, when he injected mice with virulent strain, the mice died. When he injected mice with 2R non-virulent, they survived. So therefore, after these controls, or along with these controls, he also tried whether infectious material is sensitive to heat. You know that proteins can be heat inactivated. So when he actually heat inactivated or heat kills 3S infectious strain of bacteria, of course mice survive. So this would suggest that maybe it is a protein which is responsible for pathogenesis. However, the result of the parallel experiments or parallel group of experiments were really surprising. What happened was when he mixed non-infectious 2R bacteria with heat-killed, heat-inactivated 3S pathogenic bacteria, so with those bacteria, what he found out was that mouse died. So basically, if it was protein, this would not happen if proteins were responsible for killing. This was confirmed when he actually isolated bacteria from these mice, and he found out that all these bacteria were 3S. So they were all infectious, virulent, pathogenic bacteria. So he ruled out all possibilities that the stock heat inactivation was not complete or there was mutation in 2R. So after doing different sets of experiments to rule out those possibilities, he concluded that 2R bacteria somehow acquired genetic virulence material from the dead 3S bacteria, and that this 2R bacteria has been transformed. And the substance which actually brought upon this transformation he called transforming principle. So Griffiths did not really know what the transforming principle was. He actually theorized that some substance in polysaccharide code might have actually caused pathogenesis. Many other laboratories confirmed his findings, and only later on, 10 years actually later, Avery, McLeod, and McCarthy's managed to isolate transforming principle and characterize it. So they did a set of very smart experimentation where they incubated this transforming principle, what they call transforming principle, with RNAs, which would destroy RNA, with proteases, which would destroy proteins, and with DNases, which would destroy DNA molecule. And upon treatment of transforming principle and injecting it back to mice, they saw that RNases and proteases did not do anything. That basically the 3S strain treated with RNases and proteases were still infectious and they could still kill mice. However, what destroyed the pathogenesis of transforming principle was DNAs. What does it suggest? That actually once you destroy DNA, the transformation principle or transforming principle does not work anymore. So this result were published and provided evidence, very strong evidence, that transforming principle, which actually carry genetic information, resides in DNA. The second set of experiments which clearly show that DNA is carrier of genetic information came from studies of Hershey and Chase. Hershey and Chase worked with bacteriophages, viruses that infect bacteria, and they focused on the T2 viruses. We know T2 bacteriophages compose of approximately 50% of proteins and 50% of DNA. This was important for the experiments they defined, where they labeled both proteins and DNA. So before we go to the experiments, I would like to also outline basic thing about bacteriophages. What was known at that time? It was known that bacteriophage, once it infects bacteria, it injects its genetic material into bacteria, and the code stays on the surface. Nowadays we know that bacteriophage compose of the icosahedral head, outlined right here, which enclosed genetic information, the neck, and the spikes which attach to bacteria. So we know today that it is a DNA which is injected into bacteria. However, it was not known at the time. So what Hershey and Chase did, they labeled bacteriophages either was S35 radioactive sulfur or P32 radioactive phosphorus. We know that P32 will label DNA and S35 will label proteins. So once they prepare these labeled phages, they infected bacteria, they let certain time go for infection to proceed, and afterwards they put the culture into blender to shear off coats from the bacteria. After blending, they separated the bacteria from the fluid by centrifugation, and upon centrifugation what they obtained was, in the sediment they had bacteria, and in the supernatant there was a fluid which composed of the code, bacteriophage code, which did not enter the cells, the bacteria. So now they analyze both code and bacteria. So from S35 culture, when they analyze these preps labeled with S35, what they found that Most S35 labels stayed in the fluid. It did not go into bacteria. Upon cultivation of this bacteria, they did not really detect it as 35 labeled. They didn't see much of label inside of cells. Contrary to that, when they analyzed culture labeled by P32, they saw very little in the fluid, but what they detected P32 labeled inside of bacteria which they grow. Not only that, bacteria were infected with bacteriophages and produce new progeny, and what they found out that the progeny, a lot of them carried radioactive P32. So the labeled DNA was passed from phage, which was entering, infecting from the genetic material of the phage, which entered bacteria, was carried to the progeny. So this clearly identified DNA as the genetic material responsible for carrying genetic information in bacteriophages. All the experiments we discussed and the results they yielded provided the base for the most important discovery of the three-dimensional structure of DNA by James Watson and Francis Crick. They published their work in 1953. Now, I want you to realize one thing. Watson and Crick did not generated or collected any new data. They built a model, DNA model, based on the information which were there in the field. At that time, it was known that DNA consists of nucleotides and that each nucleotide contains a sugar, a base, and phosphate. Chargaff rules were established. It was known that number of adenine correspond to number of thymine and number of cytosine correspond to number of guanines. What was not clear was how the nucleotide fit together into the three-dimensional structure and how they can actually provide base for faithful replication. Beside these discoveries, what really helped Watson and Crick to define three-dimensional structure of DNA was were studies from X-ray diffraction, and the best images were obtained by Rosalind Franklin. This is her image of diffraction pattern of DNA. This story of Rosalind Franklin is quite sad. She was working with Maurice Wilkins. Both were using X-ray diffraction to study DNA structure. But because of their personal misunderstanding or problems, they did not collaborated or worked together, but each one published their results separately. At certain meeting, Maurice Wilkins mentioned and showed the result of Franklin to Watson and Crick, and basically Watson and Crick used this pattern to define the structure of the DNA, three-dimensional structure of DNA. So basically all these discoveries made by many different scientists gave base or created the stage for defining or discovery of the three-dimensional structure of DNA by Watson and Crick. So Watson and Crick analyzed or tested various possible structures. They built different kind of model trying to fit nucleotides into three-dimensional structure. What was the breakthrough was when they realized that actually adenine base can bond with thymine base and cytosine base can bond with guanine base. Not only this explains Chargaff's observation, but it helps them to construct three-dimensional model. This model was double-stranded right-handed alpha helix with the phosphates and sugars on the outside and the bases in the interior. They published this model, their discovery, in 1953 in Nature, and this paper was very elegant, and this discovery was outlined as the most important discovery of the 20th century, and we have to agree with that. Imagine if we did not know structure of DNA, we would not be able to develop many genetic studies. We would not be able to define DNA replication. We would not be able to define transcription, gene expression, and so on and so on. So in 1962, James Watson, Francis Crick, and Maurice Wilkins, who did X-ray diffraction studies, were awarded Nobel Prize for Physiology and Medicine for the discovery of DNA structure. Rosalind Franklin, who actually contributed enormously to the discovery of DNA structure, was not awarded by Nobel Prize, and the reason was that she was dead at the time. She died of cancer at the age of 37. However, her contribution is acknowledged in most publications nowadays. So, in most organisms, DNA carries the genetic information. However, there are exceptions, and the exceptions are certain viruses, RNA viruses. It has been shown in 1956 using tobacco mosaic virus, a virus which infects tobaccos, that genetic information of these viruses could be encoded in RNA. So the genetic material can be carried by DNA but also by RNA.